All right, we're getting to our third part of our PowerPoint notes on Chapter 8, Microbial Genetics. During this section of notes, what we're going to be focusing on are looking at how gene regulation occurs by specifically looking at inducible and repressible operons. We're also going to take some time to look at different types of mutations, and then we're going to look to see how prokaryotes are able to do genetic transfer and genetic recombination, and then look briefly at the relationship between genes and evolution. So a lot of kind of a hodgepodge of things in this last section of notes. So when we talk about regulation, we have genes that are constitutive, genes that are expressed at a very fixed rate, okay? That means that those generally are genes that maybe need to be expressed all the time at a very fixed rate. Maybe there's certain proteins that we're going to need a constant supply of. But other genes only need to be expressed as needed, okay? Because it would be wasteful for your body to just make proteins that you really don't need. So we have repressible genes and we have inducible genes, and we also have catabolite repression. So one of the things we look at in prokaryotic cells is what's called an operon. And so it's important to know the different parts of an operon. First of all, you have your regulatory gene right here. You have the promoter region. Again, that's this area that signals where RNA polymerase can attach. You will have what's known as an operator, and depending on whether this is blocked or open, that will determine whether RNA polymerase can attach and go through transcription to copy the DNA code that can then be used to help make a protein. And then you have any number of genes that might be located along here. Now, when we talk about the lac operon, which is one of the most common ones, there's 3 different genes there. So the lac operon is an example of an inducible operon. We call it inducible because it's normally in the off position, so we have to induce it to be turned on, okay? And again, lac, that's short for lactose, okay? So it's normally in the off position because there will be a small molecule that binds to the bacterial regulatory protein and changes its shape so that it cannot bind to the operator, thus switching an operator on. And so when lactose is absent, that means the repressor protein can attach to that operator region and it turns the operon off. Okay, it's turning it off because it blocks the RNA polymerase from binding to the operator. Okay, but as soon as we have lactose, let's say you drink that glass of milk and now you've got lactose in your system, once that lactose is present, it's going to attach to that repressor protein and it's going to inactivate it. It's going to change its shape so that it leaves the operator. Okay, it's no longer bound to the operator region, thus turning the operon on. In other words, the RNA polymerase will be able to bind to the operator. So let's take a look at this right here. Okay, what we need to be able to do is we need to be able to allow RNA polymerase right here to travel down to transcribe these genes here. But if the repressor protein is blocking the way, that means that the RNA polymerase can't go. Okay, that means transcription can't occur, and thus you'll never have translation occurring either. As soon as you have lactose present, that's being symbolized by this little green symbol here, it's going to change the shape here of this repressor protein and it's going to remove it from this operator region here. Well, now there's nothing binding to it up in this region, so look at the RNA polymerase was able to travel down, it was able to transcribe the Z, Y, and A genes, and those genes are used then, they're translated during translation into these different proteins, and these different proteins then are going to be able to break down the lactose. And once those proteins break down the lactose, those different enzymes, and those disappear, guess what? Then this repressor protein can go right back up there and block the way, because if we don't have any lactose present, we don't need to have any of these proteins being made. Now, a repressible operon is called that because it's normally in the on position, so we need to repress it or shut it off. And the trip operon, called that for tryptophan, has a protein that inhibits gene transcription. This protein is going to bind to the DNA in or near the promoter. When it's bound to the operator, there's no production of tryptophan. If there's too much tryptophan present, it binds to the repressible operon, which then binds the operator. Okay, and then that would shut it off. So if we look at this, the tryptophan operon, it's normally on. It's going, nothing's blocking this operator here, so the RNA polymerase is able to travel along, and it's able to go through transcription. It's able to go through translation and able to make the proteins here and make the enzymes for making tryptophan. Okay? As soon as you have that tryptophan being made, now it's going to bind with the repressor, and it's going to block. It's going to activate that repressor protein, and it's going to block the RNA polymerase. Now it can no longer go through transcription or translation. Okay? So repressible operons are on, and once we make that protein acts as a binding agent to shut off. It activates the repressor protein and shut off transcription, whereas an inducible operon is usually off, and we have to induce it to be able to turn it on. And here it's showing you all this again as far as repression. Okay, so that's a little bit on operons and inducible and repressible operons. Switching gears a little bit here, if you remember from biology, a mutation is any change in the genetic material. Okay, so usually it's a change in your DNA. So most mutations are going to happen during DNA replication. They can be neutral, beneficial, or harmful. And then any agent that causes mutations on purpose is a mutagen. And then spontaneous mutations are those that just occur on their own without having any mutagen induce it. So one of the types of mutations, one of the categories, is what we call a point mutation. It's because it's a change in one base. Sometimes we also call this a substitution mutation, because all we're doing is substituting one base for another. There's two different kinds that can happen here. A transition mutation, where you exchange a purine for a purine, or a pyrimidine for another pyrimidine. Purines are guanine and adenine. Pyrimidines are thymine, cytosine, and for an RNA uracil. Okay. A transversion is when you exchange a purine for a pyrimidine, or just the opposite. Okay? If it ends up with the amino acid being changed, like it actually changed one amino acid right here, for example, we had a code for glycine, but by changing that that base, it turned it into serine, a new amino acid. We call that a missense mutation, as it's no longer the same amino acid that it used to be. A nonsense mutation is one that changes an amino acid into a stop codon. All right? So here, we had lysine as the amino acid. It was TTC, and that coded for AAG in the messenger RNA, which coded for lysine. Well, by changing this right here, this T to an A, then it then was copied in transcription as UAG. UAG is a stop codon. Well, what's going to happen? None of the rest of this protein is going to get made because you're not going to have any more amino acids being put down. So we call that a truncated protein, and a lot of times it's just going to lose its function and not function properly. Now, the other type, so far all of that was just those were just point mutations. Okay, so point mutations can be transitions or transversions, and they can result in a missense mutation or a nonsense mutation. A frame shift mutation is named that because you have either an insertion or a deletion of one or more nucleotide pairs. And when you do that, it literally shifts everything down. Okay? So if right here, okay, if we, oops, I guess they're showing it right here. If we remove that middle A here from adenine, what happens is everything shifts back this way. So now, instead of having the AAA, you would have AAC, because everything shifted back because of that space that was created, because we always have to read DNA every 3 bases at a time to make those codons. So that coded for leucine, alanine, because now you've shifted everything down. Notice how all these changed along here. Okay? So usually frame shift mutations have a much more major effect on the resulting protein than in point mutation. Not always. Point mutations can cause damage too, but really mutations don't occur very often. Again, they usually occur during DNA replication, but they're not very common. Obviously, if you're exposed to a mutagen of some sort, that's going to increase your risk of having a mutation. Those things that can act as a mutation or mutagen is radiation. Ionizing radiation such as X-rays and gamma rays can cause formation of ions that react with your nucleotides in the deoxyribose phosphate backbone. That's why you always need to protect yourself. They used to have you wear those big, lead vests when you were in the dentist office to protect you against the X-rays. Now they're a little bit better at being able to focus right at your teeth. But you notice that the dental assistant, they always get out of the room when they're doing the X-rays, so they're not exposed. All right, UV radiation. A lot of you are exposed to that, especially if you like to lay out in the sun. How this can cause damage with your DNA is that it causes what's known as thymine dimers. Well, di means 2, so it means anytime you have 2 thymines side by side, they actually start to become cross-linked, okay? And then that can kind of disrupt the normal base pairing. And so a lot of times then an endonuclease can cut those out, and then DNA polymerase fills in the gap, and then ligase seals it up, okay? So we do have some DNA repair that they can kind of help, even if you do have some of that damage to your DNA. But overexposure and lots of exposure can, over time, cause damage, and that's why it puts you at risk of skin cancer. Now, photolyases can repair thymine dimers. The problem is photolyases are only in fungi, some types of fungi, okay? So we don't have photolyases. If we did, it probably wouldn't be as much of an issue for us to lay out in the sun, because if we did have damage to our DNA, the photolyases would help repair that. But we do have some other forms of DNA repair. One of them is called nucleotide excision repair. Just like the name suggests, it basically the endonucleases just cut out that part, remove it out, and then DNA polymerase is going to fill the gap and replace it with the newly constructed DNA with the proper bases that should go down there. All right, the next topic to cover is genetic recombination. Again, just like the name suggests, it's recombining your genetic information. Well, we can talk about vertical gene transfer. That's simply when a bacterial cell divides. Reproduction between generations of cells. So it copies its chromosome, and now as it makes a new cell, that DNA gets copied. So that's one way of passing on your genetic information. Horizontal gene transfer is the transfer of genes between cells of the same generation. So we're going to talk about how we can, some bacteria can pass on their genetic information to another bacterial cell. So genetic recombination is the exchange of genes between DNA of two DNA molecules. Okay, crossing over occurs when two chromosomes break and rejoin. So we talked about that during prophase 1 of meiosis 1. Crossing over can occur when your homologous chromosomes line up in eukaryotes. In bacterial cells, they can also have genetic recombination if they get DNA from donor cells that they pick up. Maybe they brought it in through their pilus, and if that joins then with their bacterial chromosome, they can now have a new combination than what they had up here originally. Transformation is a process whereby bacteria can pick up DNA, donor DNA from their surroundings, and incorporate that into their genetic chromosome, and now they'll have some of those traits that they didn't have before. And this was first studied by Frederick Griffith when he studied these pneumococcus bacteria, and he looked at two strains, ones that had a capsule surrounding them and the other that did not. These were non-encapsulated. Well, the ones with a capsule were lethal. This is pneumococcus bacteria, so it causes pneumonia. And so when he injected that ones with a capsule, he found out that those were pathogenic. Those were lethal and caused death in the mice. When he had used the smooth strains that did not have the capsule surrounding it, or the rough strains that didn't have the capsule, those mice remained healthy. So then what he did is he heat killed these ones over here that normally cause pneumonia and kill the mice. He heat killed these and injected them into the mice, and the mouse was healthy. Then what he did is he mixed these two types. Heat killed, like this one right here, and then the ones that don't have any capsule surrounding them, these two. Well, both of those cause those mice to live normally. But when he mixed them, the mouse died. And guess what? It looked just like the kind over here. Well, how could that be? Actually, when he mixed these, the ones that did not have a capsule on it actually picked up DNA from these. Even though they were heat killed, they were able to pick up the DNA, incorporate into their own living cells, and now they had this characteristic over here, and they were lethal and caused death. So bacterial transformation is the ability of bacteria to pick up donor DNA from their surroundings, take it in through their pilus, and then those genes become part of the bacterial chromosome, and thus then now they have those new characteristics. Conjugation, that's when two microbes line up next to each other here, and through their sex pilus they can actually transfer genetic information that then again can become incorporated into their bacterial chromosome here, and now they have some new characteristics. So conjugation, it would probably be the equivalent of what we might say bacterial sex, I guess, the conjugation. All right, so it's a direct transfer of genetic information from one microbe to another, whereas transformation, bacteria are just kind of picking that up from their surroundings. Okay, it could come from a bacterial cell that was busted open and some of their contents kind of leaked out. They could pick up some of the genetic information from that bacterium. A third way is transduction, using a bacteriophage. A bacteriophage or a bacteriophage is a virus that infects bacteria. Okay, and so what they do, viruses have a protein coat and a nucleic acid core. So what they do is they inject. They are like little landing, like little, I don't know, space little ships that dock and land on the outside of the bacterial cell, and they basically inject their DNA into the cell. Well, in the bacterial cell, takes up some of this, or the bacteria, the virus actually breaks up some of the bacterial cell DNA and incorporates some of that bacterial cell DNA into its own body and starts producing, kind of takes over the cell, starts producing more and more viruses. The virus then busts open that bacterial cell, destroying it, and it will go and infect another recipient cell. And when it does that, it then incorporates some of its DNA, some of the bacterial DNA that it got from this cell over here. It's now incorporating that into that new bacterial cell. And so that's another way that bacterial cells can get new genetic information. Plasmids are also, those are those extra little pieces of DNA, and they a lot of times can carry genes that code for antibiotic resistance. So those are the small circular pieces of DNA. They're extra. They're not part of the big bacterial chromosome, but once they are taken up by a bacteria, whether it be through the pilus, then they can incorporate that and have some of those genes as part of their genetic information. So that's another way to have a little genetic recombination. Transposons, also known as jumping genes, are segments of DNA that can move from one region of DNA to another. So they can insert in different places, and depending on where they insert, they might cause some issues with the gene, if they disrupted another gene that coded for something else. So here they're kind of showing you that, how it just inserted itself here, and it became part of that bacterial chromosome. As far as genes and evolution, remember that anytime you have a mutation or a change in your DNA, or if you picked up some new DNA and are recombined in some way, that's going to provide diversity or variation. Variation is a component needed for natural selection to occur. Okay? Remember, organisms that are best fit for their environment. In other words, they're able to survive. They have adaptations that allow them to survive and reproduce and be able to pass on that trait to their offspring. Those that have the best fitness are able to survive and reproduce, will pass that on to the next generation. And that's what's needed for natural selection to occur. And so the reason we have so much variety, the reason sometimes it's tough to contain microbes, especially various kinds of viruses, they mutate a lot more frequently. And because they mutate, then it's tough for us sometimes to make vaccines or any other treatments that can take care of them, because each time they mutate, then they're a little bit different. And in some cases that might, their goal is to try to survive and pass on traits to offspring too. So each time they survive, those that are a better fit have a better chance of survival. Okay, that is it for this chapter on Chapter 8, Microbial